All right, let's go to chapter 7. Let's go to chapter 7. This is probably the most important chapter for your registry as far as safety goes. There's not a lot of meat or substance in it, bless you. That's why I just gave you a few key terms, and those are what we're going to actually talk about today. So first we want to talk about the first concept called LET. LET, I'm on page 117. LET, so this is the amount of radiation that is being transferred to the body as X-ray passes through. LET, linear energy transfer. They give you a different definition here. It can seem a little confusing when you read it. It says the average energy deposited per unit length of track. It's called the LET. Per unit length of track, they're just talking about how long or how far the radiation has to travel. That would be the track.
So, amount of radiation transferred to the body?
It's the amount of radiation transferred to the body, yeah. If you want like a simple definition, the amount of radiation that's being transferred to the body as the radiation passes through. So as the X-ray passes through, the amount that the body actually receives is called LET. So they calculate this with a formula. You do not need to know the formula for the registry, so I'm not going to teach you the formula.
The thing is tissue gray.
It is not tissue gray because LET is not a measurement, it's a descriptive term. Yep, good. LET is just a description, so it's describing what's actually going on. So you have high LET or low LET, right? X-ray is considered low LET, so low linear energy transfer. That means that when the X-ray passes through the person's body, not a lot is actually absorbed, right? So we have some indirect action that's going on because we have ionizations. We could have some cellular interaction, maybe some DNA interaction, but it's not particulate radiation, meaning it doesn't have mass. Even though X-ray has that dual particle where it's a wave or a photon, it could actually be contained as a photon or a wave. They call that duality. It is still considered low LET. So X-ray is not super damaging to the body considering other types of radiation. It gives you a little chart on page 118 to classify low LET versus high LET. The ones that you definitely want to know in the difference here on the box 7.1. You want to know that gamma rays and X-rays are both low LET. Low LET. So when they're passing through, really they're just trying to get through. There's not a lot of interaction occurring, as opposed to alpha particles, which have a weight. So alpha, remember, she's our big girl. When she passes through, she's going to do a lot of damage, interact with everybody, hang out at the party for a while before she passes through. So high LET, you want to know alpha particles. You don't need to know the neutrons and the charged particles, the ions and the heavy nuclei. I've only ever seen questions regarding high LET and low LET based on gamma, X-ray, and alpha particles. So those are your main ones. So higher LET that you have, the more interaction that you have with the radiation in the body. So they're going to stay, hang out, play, interact, maybe do some damage, and then leave. For X-rays and gamma rays, they're really just passing through. So think about them trying to get through to the actual detector. They're not hanging out. There's not a lot of interactions going on. There's actually no direct action of radiation. So it's all indirect when we talk about X-rays.
So would beta particles be low LET particles?
Beta particles. So any particulate radiation is going to be higher because we have that mass. So if we have an actual mass and it's talking about any kind of particulate radiation, you want to associate it with high LET. Good question. Good. All right. So in here, we also want to discuss a concept called RBE. RBE. So turn to page 120. Oh, okay.
I just want to make sure they're all supposed to be.
Yes. Thank you. All right, page 120, RBE. So RBE has a relationship to LET. They are directly related. So you want to write that down. I could definitely see a registry question about that. What is the relationship that they share, inverse or direct? They're directly related, meaning that when one goes up, the other one also goes up. When one goes down, the other one follows and goes down in suit. So RBE, they call it the relative biological or biologic effectiveness. So I do like this definition. It describes the relative capabilities of radiation with differing LETs, remember, linear energy transfers, to produce a particular biologic reaction. So how is the radiation interacting with the biology of the body? Not necessarily is it going to pass through quickly or fast or slowly, but how is it actually able to biologically cause damage? Is it only interacting with cells? Is it only interacting with the nucleus? Is it only interacting with DNA? How much effectiveness to the biology is able to take place? So I like to kind of summarize it in the ability to produce biological damage, if you want a shorthand definition. The ability to produce biological damage. That's going to be your RBE. Again, if it's high LET, it's going to be high RBE. If it's going to be low LET, again, follow suit, low RBE. So these terms tend to be used synonymously, but remember, think about the energy transfer when you talk about LET. So is it passing through fast or slow? LET. When you talk about actual damage that is taking place in the body, associate it with RBE. But these terms are directly related, so we want to remember that. RBE also takes into account the radiation weighting factor, so that's kind of how that got developed. They said, not all radiation is equal, meaning that if I have an alpha particle, it's going to do much more damage than just a typical X-ray or a gamma ray. So that's when somebody decided, hey, I think we need some quality weighting factors for radiation to determine how much dose a person is actually getting. 5 gray or 5 sieverts of X-ray is not equal to 5 gray or 5 sieverts of alpha particles. Alpha is much more damaging. So that's kind of where those radiation weighting factors started coming together. All right, something else we want to talk about, the OER, the oxygen enhancement ratio. This is very important because our body is composed of 2 components that are very radiosensitive. Look at that. It just kind of lights up our body to be sensitive to the radiation. One of them is water and the other is oxygen. So when there is oxygen present, the surrounding tissues become extremely more radiosensitive. So when aerobic conditions exist, when we actually have that oxygen present, the cell sensitivity to X-ray increases. So you can say increase oxygen, increase cell sensitivity to X-ray. So the more oxygen that you have present, the more sensitivity to X-ray or any type of radiation you'll have. The OER that they list in here, the oxygen enhancement ratio, you don't need to know how to actually calculate it. It is describing the oxygen enhancement actual theory numerically, so you could get a number out of it. So if you wanted to know. If you want to know, I'll write this down, this is good knowledge, the OER, or the oxygen enhancement ratio for human tissue is 3 to 1. So that means that radiation has a 3 times greater effect in our body than any kind of non-aerobic condition. So that means that we can really light that radiation up, right?
What's that called? What ratio is that?
The OER, it's on page 120. So the oxygen enhancement ratio. So the oxygen effect is just noting that when there's oxygen present, you have higher sensitivity to radiation. But the actual ratio, the OER, is just describing it numerically. So if you want to put a number to it. We know that when there's oxygen present, that our cells are more radiosensitive. So if we want to put a number by it, the human tissue OER is 3, meaning that we are 3 times more sensitive to radiation than something that is anaerobic or anoxic, if you want to say without oxygen. So you think about like this table or a dead person that doesn't have oxygen, right? We have oxygen being carried through our entire body, so we're very radiosensitive. And like I said, there's another thing that actually increases the sensitivity, and that is water. So if you turn the page to page 122, they call it radiolysis of water. Radiolysis of water. So they group these up into direct and indirect. We're not going to talk about these today. If you want to read about it, you can. That's why I gave you the worksheet. I do encourage you to read more about it. It discusses the difference between the free radical being produced. A free radical, if you haven't heard that term. Anybody heard that term before? Okay. Anybody know what it is? Free radical? What is it? Tell us.
Like toxins?
Toxic, good. Toxins, good. So it's a highly reactive and unstable substance. Highly reactive and unstable substance. It's generally caused through indirect action. So when you have X-ray that passes through your body and you have that indirect radiation happening, you have steps or events that are occurring. The radiation doesn't just go into our body and immediately take effect to the nucleus or take effect directly to the DNA channels. Unlikely. I mean, I can't say exactly where everything is in our body, so it's just a very, very low chance of that actually happening. So any damage caused to our body from X-ray is normally caused by a free radical that's produced through the indirect action. So when the radiation is actually working through the water in our body, that makes it more highly radiosensitive. Sometimes one of those H molecules or O molecules are knocked out and it produces a free radical. This is kind of the beginning stage of indirect action and how you would actually have damage in your body. The majority of radiation damage to our body is through indirect due to the high H2O contents of the cells. So you think water surrounding the cells, the cell membrane, it's all composed here. It makes us more radiosensitive. So water and oxygen are the two things that are going to increase sensitivity of the human tissue to make it more radiosensitive, meaning it could be more damaging. So turn the page to page 124. 124. We're going to talk about two different types of breaks. By chance, if we have X-rays passing through our body and it does strike DNA, it could be deadly to us or our future generations. Nobody's actually going to die immediately. It could just start weeding away at your DNA. It's not like instant death from an X-ray. So the first thing we want to talk about is a single strand break. It's on page 124. A single strand break. We don't need to know specifically the point lesion. That's where the actual injury takes place. But a single strand break. So we know that DNA is composed of a double ladder helix, right? They give you a picture on the next page, page 125, that double helix that is spiralized. We have AT and the GC that are connecting our DNA, and this is the actual genetic makeup of our body, if you've forgotten that from anatomy. So if we have radiation to strike one point on the ladder DNA, it is called a single strand break. They give you two different pictures of it here. You can have a single strand break in one rung, or you can have a single strand break in multiple rungs. But single strand break is going to just be one hit. So one hit. This is going to be our most reparative stage. DNA can repair, and if we only strike it once, it is much more likely to recover than hitting it over and over and over and over again. Just think about how fractures occur. If you hit your foot once, you're not super likely to have a fracture. If you hit it again in the same spot or multiple spots at the same time, you're more likely to have a fracture due to compression. So single strand breaks, most reparative through the body, and only being hit one time. The next type of break is a double strand break. A double strand break on page 124. So pretty easy to figure it out. Anytime that DNA gets struck twice by radiation or X-ray. So on page 125, they give you an image of a double strand break in the same rung. So the same rung or the same level of the DNA on the ladder. It's figure 7.8. A double strand break in the same rung causes complete chromosomal breakage, resulting in a cleaved or broken chromosome. This is where we start talking about chromosomal mutations. So we know that our DNA is passed on to our children, right? That's our actual genetic makeup. When you have a single strand break, it's very unlikely that you're going to pass that trait on to your children. When you get those double strand breaks that are harder and take more for your body to actually repair and restore the DNA, it's very possible that you could have a mutation take place. So a mutation on page 125. A mutation is any kind of an interaction with the DNA molecule that radiation may cause a change or a loss in the nitrogenesis base of the DNA change. So once we start changing your actual sequence of the DNA or modifying it in any way because we had a double strand break or it's trying to repair, but your body actually codes the wrong letter to the DNA and you start mutating, is where it can really get difficult for your future generations. So when we watch that video, I think we played a video about Chernobyl and how the people were kind of shunned there. They didn't want them to have children. This is why. Because if you are exposed to a large amount of radiation and you have multiple double strand breaks and mutations in your DNA, when you have children, they are going to carry those same genes and recessiveness and dominancies. So even if it skips a generation, it will come back around. So when you have some kind of a large radiation event like this, know that mutations can be consistent for many generations to come. Remember it talked about how much radiation was actually present in the milk in the fridge. So can you think about how much was actually received in the bodies of the people that were close to that event? I mean, we're water and we're oxygen. So where there's water and oxygen present, we have extreme radiosensitivity. So we want to protect our bodies as much as we can. So we have single strand breaks, easily repaired. Double strand breaks, more difficult to repair, more than one actual event in the strand breaking. And then we have mutations, which are kind of our catch-all here. We don't want the mutations to occur. This would be the last thing in the sequence of events that would happen. Can we say that mutations aren't happening when we x-ray people right now? No. But because of the low LET and low RBE of x-ray and the low dose that the patient actually receives, it's very unlikely that we're mutating the DNA. However, radiation is spontaneous. We can't choose what interactions are actually occurring in the body. We can't choose where the radiation travels. We know that it travels in straight lines, but it could hit a molecule of DNA. It could hit the target cell. So it chooses where it goes depending on the beam angulation, the part position, things like that. Most of these mutations are going to take place though through a large radiation accident or event. So do know that. All right, let's tab over to page 128. 128. Target theory. So you should have researched that a little bit when you were waiting to find your clinical placement. So somebody tell me what it is.
So it's like a random event, and like these targets are like struck in like a random radiation.
Okay, it's a random event. What is the target?
Random. For target theory?
What is the target? Yes.
The master molecule.
The master molecule. What is the master molecule? DNA. Good. DNA. Good job. So DNA is the master molecule because it is our makeup. So DNA, and specifically it is the nucleus of the DNA cell.
If they receive multiple hits, it'll be like inactivated or the cell function will die.
Exactly. So the master molecule, so DNA, if the DNA is struck, specifically the nucleus of the DNA, that master or target cell theory states that if it is struck, instant death will occur. So if we kill the cell, it's non-functioning. So the target theory is completely random. Again, we can't choose exactly where the radiation goes. We have no control over that. We do have control over collimation, but the theory is that if the molecule is struck and if the actual nucleus is struck, then instant death will occur. We don't know that this is actually true. It's a theory. That's why they call it target theory. But DNA is the master or the key molecule. So this is the thing that we want to protect and preserve the most within our body. Where is DNA located in our body? Everywhere. Yeah, we don't know, right? So we can't say our DNA is located all right here, so just don't x-ray this part, right? It's all over our body. So you don't know what types of DNA are going to pass down to your future generations or children if you are going to have children. Know that this is just as deadly for men as it is for women, because when you create a baby, both of the DNAs involved are increased. So we pull a little bit from both of our parents there. Target theory may be used to explain cell death and non-fatal cell abnormalities due to radiation theories. So exposures that are lengthy or large in dose radiation accidents, whether it's a nuclear bomb or some kind of nuclear power plant explosion like we read about, it may actually be used to describe what's going on if there are abnormalities or if there is instant cell death. So the master molecule is essential for cell survival. So your DNA is needed, right? It's the only thing necessary in our body cell-wise for us to function completely. All right, let's go to page 131 and talk about the effects of irradiation on the entire cell. The effects of irradiation on the entire cell. I think this is the last part of our chapter. Yeah. All right, so the first one is instant death on page 131. Again, we're not talking about x-raying somebody and then immediately dying on the table. We're talking about cellular death, okay? Very, very small cellular death. This is generally used to describe a large body dose. So a large body dose is what you want to associate this with. Instant death of the cells. It gives you a number here, approximately 1,000 gray in a period of seconds or few minutes. That's a ton of radiation. So you want to know the number, and you want to know that typically instant death is associated with a large body dose. Large body dose. All right, tab down a little bit. We're going to talk about reproductive death. I like to write failure beside this one. Failure, because it's really not reproductive death, it's reproductive failure. So this doesn't affect the actual function of the cell other than if it's a sex cell, and it loses the ability to procreate. So basically the cell is sterile. So the cell is sterile. This ranges anywhere from an exposure of 1 to 10 gray. So you want to highlight that one, know that one for sure. So if a cell receives 1 to 10 gray, they could potentially not be able to procreate or reproduce the cell. Why is this important? Why is it important for our body to be able to produce cells?
For healing.
Regeneration, healing, good. Lots of functions in our body. So anytime you get a cold, if your cells get attacked, white blood cells, red blood cells decrease. Maybe you've got some kind of junk in your lungs and you need some more cells to be amped up. Your body can reproduce them and procreate. If one dies, one of the other ones steps in and pushes a new one out, right? So it gives a piece of it.
No.
May I have your attention, please?
Let's see if
May
it's
I
showing.
have your attention, please? There has been a fire emergency reported in the building.
Really?
Please evacuate the building by the nearest exit.
All right, let's see back
Do
again.
not use the elevator.
All right, let's finish up. Let me play music. All right, let's finish up. We're still on page 131. So we talked about reproductive death and the loss of the ability to procreate. Let's talk about the next one, which is apoptosis. So this is a cell that dies without dividing. So remember we talked about, or had you guys research at least, that meiosis versus mitosis in the cell chapter. Hopefully you did read over that. So when the cell is in interphase, they also call this interphase death. Interphase death. So before the cell actually divides, it ends up dying. So before the cell before the cell initially divides through any of the phases, it dies. So they also call this interphase death. Interphase death, programmed cell death, or apoptosis. Apoptosis. All right. And then let's look at the last one, which is mitotic death. Mitotic death. So this is when a cell goes through one or two of the phases of mitosis, and then they actually die. So less radiation than interphase death is required because they're dividing. So think about them being smaller cells. So as they're smaller, less developed, less specialized, more immature, they are more radiosensitive. So maybe they've gone through the first or the second phase of mitosis, and then they die. They call this mitotic death. Mitotic death. And if a cell is exposed to as little as 0.01 gray, which you might receive that when you're getting an x-ray or a couple of x-rays stacked back to back, they could experience a mitotic delay. Mitotic delay, which means that the length of the actual phase could be increased or decreased, or the actual ability to phase out or separate or split into a different phase might be delayed. They call this mitotic delay. So it still can actually divide. It has the ability to. It will get there. It just might be delayed. Mitotic delay. All right, and then the last thing we want to talk about in this chapter is on page 133. It is the law of Bergenet and Tribondeau. So this is a French radiologist and a French physician. So Bergenet is the radiologist, Tribondeau is the French physician. These two kind of grouped up and observed their recordings and findings based on radiation to testicular germ cells of rabbits. So they took rabbit testicles and radiated them, right? It sounds kind of crazy, but they actually found a lot of information. So their findings basically correlated radiosensitivity to these germ cells, so to the actual sex cells that we're producing, and they came up with this theory. It's on the very bottom of page 133. So it states that the radiosensitivity of cells is directly proportional to their reproductive activity and inversely proportional to their degree of differentiation. So the more differentiated a cell is, if it is old, say if it's a nerve cell, it already went through the stages of being a stem cell. It decided that it was going to be a CNS, and then it progresses to a nerve cell. It is much more developed than a stem cell in a baby's body or in the umbilical cord or in a newborn. Much more developed means less radiosensitive. So before they figure out what they want to do, when they're trying to figure out who they're going to be when they grow up, they're very radiosensitive, less developed, less specialized. It also has to do with the mitotic activity. So it talks about their reproductive activity. So specifically, our goodies, right? The things that make the babies. These are highly, highly radiosensitive. So the ability to reproduce is directly related to your radiosensitivity. So
134.
133, 134. We're still talking about the low of Bergenet and Tribondeau. So directly relational or proportional to their reproductive activity. So if it can reproduce, if it's a reproductive cell, highly radiosensitive, and inversely proportional to the degree of their differentiation. So if they are very unspecialized, if it's a new cell, they are very radiosensitive. So they're opposite, inversely proportional. If it is like a nerve cell, like I said, very developed, very specialized, it is less radiosensitive. So turn your thumbs opposite. Inversely proportional to the degree of differentiation or specialization, if you want to call it that. and directly proportional to their ability to reproduce, so the reproductive activity. This is kind of what they figured out here, based on older rabbits or younger rabbits, but they did their studies on testicular germ cells, so you need to know that. You need to know what they did their study on and what they found. Inversely proportional to the developmental aspect, directly proportional to the reproductive activity. That is all we are going to cover in this chapter. The rest of the chapter goes through some actual effects if you were to receive radiation to different types of human tissue. So it talks about blood cells. If you turn the page even further, it talks about ova, epithelial cells, muscle, nerve tissue, reproductive. You don't need to know the specifics of this and the effects of radiation. However, you do need to know acute radiation syndrome specifically. So in the next chapter on chapter 8. So that's what I'm gonna have you do for homework.